Our first speaker of our second morning session is Dr. Bernardo Sabatini from across the river, Harvard University Medical School. And the title of his talk is Co-Release and Integrations of Neurotransmitters in the Basal Ganglia. Welcome.
Okay, thanks for the invitation to speak, and also thanks to everybody for coming. I'm sorry, but I'm actually not going to talk about co-release of neurotransmitters. I'm going to talk about development. Yeah. Well, I'm happy to tell that story another time. I'm going to talk instead about the development of the basal ganglia and the process of activity-dependent synapse formation in this circuit. And I hope you don't mind. I'm going to stand over here because I find over there I always strain my neck when trying to talk in this room. So this is work that was initiated by Jenya Kozorowiecki and R.P. Saunders when they were a postdoc and a graduate student lab, and then was continued on by Jenya and then by a current postdoc in lab, Rui Peixoto. And this has to do, as I mentioned, with the activity-dependent processes that govern the formations of synapses between the cortex and the striatum. And so my lab studies mostly the development, function, and regulation of the basal ganglia. As you all know, these are evolutionarily old and phylogenetically conserved subcortical structures that are very important for triggering coordinated sequences of motor actions and for promoting the repetitions of behaviors that lead to reward. And you just heard Richard talk about the VTA and dopamine and so forth and its possible role in shaping animal behavior. It's quite important for shaping human behavior as well, as we know from diseases of the basal ganglia such as Parkinson's, Huntington's, and drug addiction. And so we're used to usually thinking about the basal ganglia as mediating the kinds of behaviors that we see here, which are learned motor actions that are carried out in a particular sensory context to either achieve a specific physical reward, such as food here, some more sort of esoteric sort of feeling of pleasure, or in this case, the direct euphoria associated with administration of drugs of abuse, IV. But there's really been a case building for a while now that the basal ganglia may be quite important for some diseases in which they haven't traditionally been associated, for example, autism. And so if we think about many of the main symptoms of autism, such as difficulties in language, these perturbations of eye contact, repetitive motions, behavioral inflexibility, perturbed social skills, these are all things that one can relate to the basal ganglia. For example, there's very clear studies in humans of activation of the caudate in generation of language, which after all is a goal-oriented motor sequence. One can think about it that way. There's direct innervation of eye gaze fields by structures that are output structures of the basal ganglia. This is well studied, of course, in primates and so forth. So one can really make this case. And so I want to give you some evidence of the kinds of perturbations of activity-dependent processes in the development of the basal ganglia that we think may be related to some of the symptoms that are here, although obviously I won't make a direct link in humans today. These are going to be mouse studies. And there are, though, links, as I mentioned, being made within the human literature that I'm not going to go into today. The idea basically emerges that there are loops that exist between different areas of cortex, but also between different areas of cortex and the basal ganglia. And it can be the over-strengthening of these kinds of recurrent loops that might underlie, for example, stereotypy, the repetitive motions again and again without clear reward. And so we wanted to understand what are the factors that govern the development of these kinds of recurrent loops, and I'll tell you more about them in a minute, and probe the idea as to whether early life perturbations of activity could lead to propagating changes in these circuits that might underlie some of these behaviors. And these are cases that have been made already in the literature, studies of mice trying to relate this to autism genes. And I should have put up this here, a study from our keynote speaker, Tom Sudoff, with Rob Malenka, also exploring this process in neuroligand function and repetitive behaviors and motor learning. Okay, so we study the mouse because there's rapid maturation that occurs in the time scale of a few weeks. And so the first step really was to understand where in this process does the maturation of the striatum, the input stage of the basal ganglia, occur. So from a rodent perspective, I'm going to give you a very simple cartoon of the basal ganglia that we'll use to frame today's talk. The input stage of the basal ganglia is the striatum. This is a very large subcortical nucleus. It receives glutamatergic inputs from cortex as well as from thalamus, although we won't discuss this today. And then it sends two inhibitory output pathways. One is the so-called direct pathway by which the striatum inhibits the substantia nigra reticulata, reticulata in turn inhibits thalamus. And so here we have two GABAergic projections that cascade. And so the net effect of this pathway is actually to excite cortex. And it's thought that activity in this pathway promotes the generation of movements, at least locomotion in an open field task, and also is somehow in itself inherently reinforcing, although we don't understand the mechanism of that. This pathway is counterbalanced by the so-called indirect pathway, in which there's an additional GABAergic synapse in the globus pallidus, and then back to reticulata. Here we have 3 inhibitory synapses, and then cascading excitation. And so the net effect of this system is actually to inhibit thalamus, withdraw excitation of cortex, and therefore reduce cortical activity. And this pathway is thought to be involved in switching motor action, inhibiting motor action, and again, the mechanisms are still there, somewhat unclear. This whole system is under the tutelage of midbrain dopaminergic centers. We just heard from Richard, as I said, about VTA. Substantia nigra compacta also produces dopamine and heavily innervates the motor regions of the striatum. And in ways that we don't understand, the release of dopamine within the structure will promote the repetition of actions that have been associated with reward. And so we can think of this whole thing as basically these 2 loops: direct pathway counteracted by indirect pathway of 2 different signs, and some kind of judge that imparts value on the system through the substantia nigra. So I want to contrast just a few funny things about this circuit. One is that we have cascading inhibition. Okay? Secondly, not drawn here, there is some topographic organization of this. And so it's very interesting to think about what kinds of rules could govern the formation of a topographic organization through cascading inhibition. It's not really obvious how to do that. And most of the studies of development have been carried out in systems like this, in which, in a very simplistic sense at least, we can think of the eye as sending glutamatergic input into thalamus and so forth into cortex. And the story has been emerging over many years now that, for example, waves of activity within the eye can propagate information about nearest neighbor relationships into thalamus and so forth into cortex. And this sort of cascading excitation can be used to pass along the kind of information that can be used to establish local wiring rules, for example. Okay? Similar things occur in the auditory system. Right? So what about this other system, which we have cascading inhibition? What are the kind of rules that govern the wiring of that? So I'm going to tell you about 3 kinds of assays that we use today. This is a striatal projection neuron that's been filled with a red fluorophore through a whole cell recording pipette, so we can see the structure of the dendrites of this cell and spines that are located on it, dendritic spines. And so today we're going to analyze synapses in 3 ways. One is simply by morphology, counting the number and density of dendritic spines. The second one is by recording spontaneous miniature excitatory postsynaptic currents, shown here, which are mediated by the opening of postsynaptic aproreceptors, indicating the spontaneous fusion of a vesicle. And this is another readout of numbers of synapses. And lastly, I'll show you some evoked synaptic responses with channel adoption that we'll get to in a minute. And the reason I'm spending some time here is that, as you'll see, these studies are done in sort of complicated genetics. And so I don't actually have all 3 of these for all kinds of data that I'm going to show you today. But when we have multiple of them, they always give the same answer. Okay? So I'm going to move between all of these as a way of sort of judging the strength of connectivity between cortex and striatum. Okay, so first, how do these projections from cortex to striatum develop? So this is a mouse that expresses Cre recombinase in a back transgenic under the RBP4 promoter. Okay? And it turns out that then this mouse expresses Cre recombinase in layer 5 pyramidal cells, including those that project the striatum. Okay? So if we cross that animal with a flox channel rhodopsin animal, we can now have channel rhodopsin expressed in corticostriatal neurons as well as in other cortical projecting neurons. But when we cut a brain slice, we can selectively activate those corticostriatal neurons. And so in fact, if we do cut a brain slice and obtain a whole cell recording from a spiny projection neuron within the striatum, we can then look at a variety of different ages, what's the strength of this optogenetically evoked current in those cells. And so you can see that at P6, there's a very small transient that's there at minus 70 millivolts, consistent with activation of a small number of AMP receptors. And this sort of grows over time to where by P14, there are many nanoamps of current that can be evoked here. Okay, so we can use channel rhodopsin to probe the development of these excitatory synapses in this ex vivo analysis of corticostriatal transmission. And we can therefore define a window over which corticostriatal transmission develops. And as I said, we've done this also by analyzing miniature EPSCs and spines. And the window really looks the same, which is that you can see this rapid increase in the strength of corticostriatal transmission that occurs in this sort of P8-9 to P12-13 window. Okay? And in fact, by the time the animal reaches P18, its strength of corticostriatal synapses are the same as around P30. Okay, so very rapid sort of explosive growth in this projection that occurs. Okay. And so first question is this actually driven by activity? Okay. Does the activity of corticostriatal neurons actually set the pace of synapse formation in this downstream structure? And the reason that we thought this might be true was because of a study we had done a few years ago in which Hongbae Kwan, a postdoc in the lab, had shown, in this case in cortical 2-3 pyramidal neurons, but I'll get the striatum in a minute, he had shown that by photoreleasing glutamate, here by a 2-photon process where he could break down a caged inert version of glutamate into glutamate, okay, that by focally releasing glutamate at this spot where there was no spine, under conditions that maximally activated NMDA receptors, he could grow a spine very rapidly in about 20 seconds. And there's a great deal of study that he did to prove that this was associated with a new functional synapse. But the point was that glutamate was a minimal signal that we could use to induce synapse formation, suggesting that the pace of neurotransmitter release by axons that innervated downstream structure could actually set the pace of synapse formation in that downstream structure. And I'm not going to show you the signaling work analysis that he did, but this was a process that was dependent on activation of NMDA receptors as well as protein kinase A in the postsynaptic cell. So based on that, you can imagine now that this loop in which we have reinforcing activity that comes through, we could have many different factors that govern the activation, the patterns of activity in corticostriatal synapses, corticostriatal projections, and therefore govern the pace of formation of synapses here. So we thought we could exploit the structure of the loop to perform genetic manipulations that would alter the activity of this circuit indirectly. And so, for example, if we think about essentially cutting the direct pathway here, which we can do by silencing the release of neurotransmitter from neurons that form the direct pathway, if we withdraw this loop, we'd be withdrawing excitation from cortex and therefore weakening dampening the activity of corticostriatal neurons. And if our idea is correct, this hypoinnervation of that projection should inhibit synaptogenesis in the striatum. And conversely, if we cut this pathway, withdrawing this inhibition of cortex, we should superactivate cortex and possibly drive corticostriatal synapse formation more rapidly in the striatum. And so RP and Genya did that already a few years ago using recombination of the VGAT gene out of either the direct or the indirect pathway. VGAT is the protein that moves GABA from the cytoplasm into the vesicle and therefore is necessary for GABAergic release from these synapses. And you can see if we silence the direct pathway creating a hypoactive cortex, we lose synapses. And if we silence the indirect pathway creating a hyperactive cortex, we can gain synapses at around P14 in that sort of right at that sort of end of the tail end of synapse formation in the striatum. Okay, so this supported the idea that, that corticostriatal transmission could set the pace of synapse formation. And just as a side, we did a little bit of behavioral analysis of these mice. When we silence the direct pathway, these animals are actually quite runty. They die by around P22 of age. But if you put them in locomotion, if you simply look at their locomotion in an open arena, you can see they move around more slowly. The indirect pathway animals, which also die sort of around P22, around the time that these animals would have to be able to forage and live independently, these guys are hyperactive in the arena until they die. And this is exactly what you expect from the model that I presented. But it says that early in postnatal life, the gross circuitry and function of the locomotor basal ganglia are in place, and these antagonistic opposing effects of direct and indirect pathway are visible. Okay, so we're suggesting then that the patterns of activity in corticostriatal synapse projection neurons are what's dictating the activity here in striatum. So, but are these two structures really active in these early days, and are they really connected? Can we see functional evidence for that by electrophysiology? And so Rui did some really difficult analysis, which is that he performed in vivo multi-electrode array recordings, silicon multi-electrode array recordings in cortex and striatum of mice as young as P10 that were awake but head restrained. Okay, so they were kind of sitting there. He could put electrodes in their brain, and he could through one craniotomy record first in motor cortex and then pass through and record in striatum, so you get activity of both structures. And so what you can see is that here at P10 he can find some spiking units, within cortex, and again, in striatum he can as well. Activity patterns are quite low. By P14 there's much more robust activity in cortex as well as in striatum, and you start to get sort of bursts of activity that are there. And I'm not going to show you the full analysis here, but you can see in these plots where each neuron is, each dot is the activity of an individual neuron, at a variety of different postnatal ages, you can see this sort of increase in activity within motor cortex over P10 to about P15, P16, as well as in striatum, and bursting patterns and so forth change that I won't tell you about it today. So both structures are sort of coming online around the same time with this increased activity. Can cortex actually drive striatum at that time? So what we did at this point is do the same kinds of experiments, but now using the same animal that I showed you before, Cre recombinase in layer 5, channel rhodopsin now expressed in those cells, and then flash light and can he drive activity in striatum by activating cortex in the same preparation? And indeed, you can. And so here's a series of 10 pulses of light delivered to cortex, and you can see that in striatum, on the first pulse you get a big activity, and then it sort of dies away, but that we can reliably cause spiking in striatum by activating cortex at this age. Now, it's kind of interesting that this channel rhodopsin stimulation of cortex drives spikes in striatum at P10. It's actually equally effective at driving spiking in striatum at P10 as it is at P14. Remember, I showed you before, though, this sort of explosive growth in the strength of connections between cortex and striatum. Nevertheless, it doesn't translate into one being able to more strongly drive the other structure at this age. And just as an aside, I'll tell you why, which is that young striatal neurons are actually hyperexcitable. And so we did some measurements of intrinsic excitability of these neurons simply by using current steps and measure spiking. And what you can see is that there are several things that make these young neurons more active, more excitable actually than old neurons. The first is that the resting potential of these cells drops dramatically between P10-11 and P13-14. And so anybody who's recorded in striatum or done whole cell recordings of striatal neurons knows that these cells are famous for sitting very, very hyperpolarized. And that seems to be a feature that actually emerges during this window that we're talking about. But furthermore, there's just a simple consequence of input resistance. If you look at a current voltage relationship, you can see that this is a very steep relationship at P10-11, becomes more shallow over time, showing, which is simply a consequence of these cells probably becoming larger and their input resistance dropping. And so therefore, if we look at spike firing versus current injection, you can see that actually it's easier to fire spikes with, let's say, 100 picoamps of current in young neurons than older ones. And these things together make young striatum very sensitive to the activity of cortex in a way that's not true later. And we think that this is probably sort of a feature that allows that structure to pay attention to the small amounts of activity that are happening in cortex. And as that activity ramps up, the sensitivity of the system actually goes down. Okay, so we've shown that these two structures are active, that they're developing, they're in this time point. Can we really make the point that activation of cortex really drives the formation of synapses in the striatum? So again, we use this animal with channel rhodopsin, these layer 5 cortical projection neurons, and now implanted an LED on the skull of this animal in an acute surgery, and then gave a relatively modest pattern of activity in cortex over 1 hour. And so in 1 hour, we delivered 300 pulses of light. They were patterned in this way, 5 pulses, and then the next minute, another 5 pulses. So one little burst of 5 pulses every minute for an hour. That corresponds to about a 10% gain in activity of cortex with the recordings that we looked at before. But of course, now it's highly synchronized compared to the more desynchronized patterns that we saw before. Then we can cut a brain slice and simply compare the strength of corticostriatal transmission here in the stimulated side versus the unstimulated side with whole cell recordings. And what we find is a very large strengthening of transmission between cortex and striatum in the stimulated side as opposed to the unstimulated side. And I apologize for skipping the quantification here. You'll have to trust me, but this really is quite representative. And so this is 300 pulses over 1 hour, synchronized, large increase in transmission. Okay. And by the way, as an aside with what Richard said, this is completely dependent on activation of PK via D1 receptors or A2A receptors, depending on which cell class you're looking at. Okay, so acute increase in corticostriatal rapidly strengthens corticostriatal pathways. Okay, so far I've been talking about this system here, manipulations here can drive striatum. I've told you a little bit about how recurrent pathways can do the same thing. But what about here? What actually data do we have about dopamine? So remember that I told you before that this process of glutamate-induced synapse formation is dependent on protein kinase A. And as I just alluded to, some of the most important targets of dopamine within the striatum are actually regulators of PK. So for example, the direct pathway neurons are recognized molecularly by their expression of type 1 dopamine receptors, which of course are positively coupled to adenylyl cyclase and to the production of PK. In contrast, the indirect pathway neurons, sorry, there's an extra letter there, these are characterized by the expression of the type 2 dopamine receptors, which are negatively coupled to adenylyl cyclase and therefore would downregulate PK. Okay, so we have dopaminergic projections of the striatum. It'd be natural to think that the activity of those projections would send a signal, right, that should possibly promote the formation of synapses in an important context, for example, for reward. And so are those synapses there and functional at an early age? Well, this is an animal, this is an image of the expression of TD tomato in an animal that expresses Cre recombinase under the dopamine transporter promoter. This is a knock-in in this case, crossed to a conditional TD tomato. So we can see red expression in the cells that express this dopamine transporter. And you can see that in SNC at birth, we already have activation of this. And furthermore, if we look in the striatum, we can see the axons of these cells innervating striatum. So even at birth, these fibers are already in striatum. And by 10 days of age, if we do a stain for tyrosine hydroxylase in nigra, we can see very robust expression of tyrosine hydroxylase in these cells. In striatum, you can see the green cells here are actually the indirect pathway that are labeled by a GFP line, but they're now surrounded by tyrosine hydroxylase positive fibers that are coming from SNC. So there's a potential source for dopamine there. So do these things interact, glutamate-induced synapse formation and dopamine activation within the striatum? And so here's an example of a striatal projection neuron at around 14 or 15 days of age, in which Genya did glutamate inclusion at this spot, and you can see that she could grow a new spine. Okay? So this process of glutamate-induced new spine growth can occur within the striatum. And furthermore, as I mentioned, it is sensitive to the activation of D1 receptors. And so here, the way we typically do the experiment is to probe the efficiency of growing a new spine when we use a short uncaging pulse of just 500 microseconds, a longer one of 2 milliseconds, or a really long one of 4 milliseconds. And so this releases more and more glutamate, and you can see that with more glutamate activation, the probability of growing a spine goes up. And now if we activate D1 receptors with SKF in vitro, you can see that we can make this process more efficient. Okay? And this is PK sensitive. So there is an interaction between glutamate and type 1 dopamine receptors and PK to determine the formation of new synapses. Okay, so we did the same kind of experiment in vivo now. Took a young animal, injected it with SKF, waited an hour, and asked how strong are synapses within the striatum. And here I'll just show you some examples. An hour after injecting SKF, we can see a large increase in the density of dendritic spines selectively on the direct pathway neurons, which express D1 receptors, as well as an increase in the frequency of miniature EPSCs. So turning on that pathway is sufficient to drive synapse formation. Now remember, this is an awake animal. And there's an interesting thing that happens when you give an awake animal an injection of a D1 receptor agonist. It runs like crazy. Okay? So this is actually really coincident. In terms of running, we're activating cortex, driving glutamate release in striatum, but providing a D1 receptor tone. Okay? So this exploration, experience, and reward, we think, can converge with this cortical drive in the striatum to dictate the pace of synapse formation. And the last thing that I'll tell you before I wrap up is a small connection that we're making to one of the disease genes associated with autism, and this is SHANK3. And so SHANK3 is a protein that's very highly expressed and very highly found in the postsynaptic density and interacts with many of the players that we heard from before from Rick, and that I think we'll hear again from Mary Kennedy. And so this is a protein that's found there. It's a major component of the postsynaptic density, and it's been associated with autism because Phelan-McDermott syndrome, which is a syndromic form of autism, is caused by a deletion of a large region in 22q that includes SHANK3, and it's thought to be the loss of SHANK3 that's really causal for the symptoms of that disease. And furthermore, there are truncation or deletion missense mutations that are being found in SHANK3 individuals with autism, but also with intellectual disability and schizophrenia and so forth. So it's not really autism specific. And Guoping generated SHANK3 mutant mice a while ago and came to the conclusion that there was hypoinnervation of the adult striatum. Okay? And this feature of repetitive grooming, which seemed to be a stereotypy there. And so our model would predict hypoactivity in cortex might actually lead to this hypoinnervation of striatum. But it's hard already to imagine how this hypoinnervation of adult striatum might somehow be responsible for this kind of repetitive grooming, which I suggested earlier would be sort of a recurrent overactivation of the circuit. So we had a big surprise, which is that when we recorded from these knockout mice, around P13, P14, we found that in fact cortex and striatum of these mice were quite a bit more active than of their wild-type litter mates. So completely the opposite of what we had expected based on our model. Okay, there was hyperactivity with increased bursting of these ages. And now, in fact, when we cut slices from these animals and recorded in the striatum, we didn't find hypoinnervation. We found hyperinnervation at early ages. Okay, so at P10, the wild-type and the knockout animals had about the same frequency of miniature EPSCs and of evoked channel rhodopsin currents. By P14, the knockout had a little bit more, stabilized at around P30, and then at P60, we actually got exactly the same result that Guoping did. There's actually fewer active synapses in the striatum at P60, but there are more early on. Okay? So there seems to be an early hyperinnervation that, for then reasons that we don't understand, later on translates into a loss of transmission there. Okay? So in that sense, this is consistent with our model because there's overactivation of cortex and there's overinnervation of striatum early on. Okay? So is this kind of overactivation of cortex really sufficient to drive those changes in striatum? So we took an animal that has this conditional allele of VGAT that's necessary to make GABA, injected Cre-encoding AAV into cortex, so we eliminated inhibition in cortex to make it hyperactive. Here you can see that the Cre-injected cortex has these really sharp waves that are due to activation of many neurons at the same time. And if you just simply look at a power spectrum here, it's very clear this perturbed activity that we can see in cortex in the injected animals. And in fact, overactivating cortex in this is sufficient to create more synapses within the striatum. This continues a little bit different from the SHANK3, continues to be present at P30, but by P60, once again, the differences are gone. So we think that overactivity of cortex can explain the phenotypes in the SHANK3. Furthermore, they're necessary for the phenotypes because we can do the converse experiment, which is to use a chemogenetic approach using HM4D to inhibit the activity of cortex, specifically of RBP4 positive neurons in cortex in these SHANK3 knockouts. And so we did this by injecting an AAV coding HM4D at P3 and then injecting the ligand CNO into these animals from P10 to P13 and then doing ex vivo analysis. And you can see that now we can reduce the exuberant corticostriatal synapses simply by shutting down cortex. And so we think lots and lots of these changes that one sees in striatum in this model, and we think in many other models, are actually secondary to alterations in the pattern of activation of cortex. So I'll stop there. We're now moving towards trying to understand whether we can rescue and perturb behavior. And so we're simply establishing conditions in which we can do analysis of grooming behavior in these young mice. This is a P17 mouse, I think, and we can obviously detect grooming. And so now we're looking at whether we can establish a causal link to behavior. So let me end there. So our conclusions is that the activity of excitatory axons can guide the pace of synapse formation in downstream nuclei. This allows for, I think, two different modes of wiring. One can be sort of a self-guided wiring of closed loops through multisynaptic circuits. And so, for example, the direct pathway can essentially wire itself because when it finds its right targets downstream that overactivate cortex, that'll feed back into striatum and stabilize itself. The indirect pathway presumably has to use different rules. Furthermore, there's sort of an active form of synapse formation where corticostriatal synapses can be, their formation can be promoted by dopamine in a PK-dependent manner and NMDA receptor activation. These processes are sensitive to both transient and chronic changes in cortical activity, and we think that SHANK3 mice actually have overactive cortices early in development. This drives hyperinnervation of the striatum. We think this could set in motion hyperconnected recurrent loops that might underlie some of the repetitive grooming and so forth those mice see. Okay, and that's currently what we're pursuing now. I mentioned the postdocs and grad students along the way that did the work. We have really a tremendous technical support staff in the lab. Not all these people are there anymore that helped in making these things happen. These are electrophysiological recordings in triple and quadruple transgenic animals that have been injected with viruses and so forth. So if it weren't for them, we wouldn't be able to do these studies. And this was funded by principally a Nancy Lurie Marks Foundation grant. Okay, thank you. Sorry, I went so fast.
No, that's good. We love speakers.
Yeah.
So Bernardo, I think it's very compelling, the idea that you have an overactivation of the cortex and the But I was just wondering, given that you see this corticostriatal neurons during development. Whether this doesn't work anyway.
I'll repeat it, don't worry.
Whether the SHANK3 mice have changes in the intrinsic excitability that actually could also be responsible for the same phenotype as you see.
That's right. So Tom is suggesting that there may be additional effects due to changes in the in the intrinsic excitability of striatal neurons in the SHANK3 mutant that might contribute to this effect. So indeed, we have to formally examine that possibility. And so we're doing recordings in the SHANK3 mice of all the properties that we possibly can of striatal neurons to look at that. So far, it looks like that's not the case. The striatal projection neurons actually look relatively normal in these early ages. The other thing I'd point out is that we can do this manipulation of reducing activity in cortex and essentially correct the phenotype in terms of numbers of synapses in the striatum by simply manipulating cortex. And so I think that suggests that the cortical drive is the primary, but there may be other effects too, and we're trying to look at that. Of course, SHANK3 is expressed in striatum. Presumably it's doing something, and we're trying to find out what that is. No?
All right, now can I ask a question?
Yes.
So the cortical instruction to the synapses onto the striatum synapses, they don't really differentiate direct pathway neurons versus indirect pathway neurons. When there's a hyper or hypoactivity from cortex, all synapses are going into one direction, or there are some sort of differentially regulated rules in terms of synaptic density or mini response in terms of indirect versus direct pathway neurons?
Yeah, so that's a very good question, and the answer depends a little bit on which experiment. So for example, when we silence the indirect pathway, which leads to an overactivation of cortex, we see exuberant numbers of synapses in both direct pathway and the indirect pathway. So it seems that increased cortical drive at that point is sufficient to increase synapse number in both. Okay. If we give a D1 receptor agonist, then we only see induction of synapses in the direct pathway. To see induction of synapses in the indirect pathway, we have to activate a G-alpha S coupled GPCR on those cells and increase glutamatergic drive, right? So if you turn on cortex very strongly, you can cause synapses to grow in both. If you want to do it in this more context-dependent manner, then it depends exactly on which GPCRs are active, and then you can start to break these things apart. For the SHANK3, we don't know if there's more synapses onto the direct pathway versus the indirect pathway, because frankly, the idea of putting a fifth allele in there to be able to distinguish the direct and the indirect pathways is a little bit, I think, too much. And so I think we're going to leave it at now, we don't see any evidence in the data for clearly sort of bimodal, like the direct pathway is hyperinnervated. But there's so much smear here that it might be very hard to pick that out.
Okay.
So, Bernardo, I'm wondering about the link of autism-linked genes to the activity-dependent remodeling of synapses. So with this mechanism that you described about the glutamate-induced spinogenesis, do you have evidence that loss of SHANK3 actually changes that or other types of synapse formation that can be induced? So that would just kind of link autism genes to local activity-dependent regulation.
Yeah, so the model we're proposing is sort of explicitly a non-local one, that it's really defects on cortex. We haven't looked at glutamate uncaging-induced synaptogenesis in a SHANK3 knockout medium spiny neuron, okay? And we could do that. We don't have any reason to believe that it would be perturbed, but we could look at that. I was trying to more build the model that many of these genes that are implicated in autism, many of which, of course, cause epilepsy as well, could be changing patterns of activity in cortex and therefore going downstream. So, for example, Dravet syndrome, mutations in the sodium channel, that seems to reduce cortical inhibition. That's exactly what we did with the VGAT flox allele. One would expect would create the same kinds of things. But yeah, we don't, as of now, we have no cell autonomous function that we can attribute to SHANK3 expression in spiny projection neurons, and I don't mean to say that it doesn't exist. We just haven't looked at it that heavily, and we haven't found it yet.
A simple question, like with this kind of treatment, do you observe homeostatic?
Yeah, so I often get asked that question, and we don't. I'm not really sure what they would look like, right? I mean, everything I've shown you in some sense is non-homeostatic, right? I say, okay, you introduce activity and you drive more activity. My guess is that very early in development, that the homeostatic mechanisms aren't really kicking in, because I sort of favor a model in which cells can become introduced into a circuit somewhat gradually, right? And you wouldn't actually want every neuron in the striatum to be active at the same rate, right? I like the idea of it kind of gradually forming the right classes of synapses, and as those synapses form, becoming more active and so forth. And so we haven't seen evidence for homeostasis here. It's possible that that change from P30 to P60, where the synaptic strength drops, that is actually an effect of homeostasis, and we need to figure that out. We're not exactly sure how to do that experiment.